So with that, I'd like to invite Dr. Niyati Jhaveri from Akoya Biosciences. Niyati received her PhD from the University of Southern California, where she worked on identifying novel therapies targeting cancer stem cells in glioblastomas. So she has extensive experience in spatial omics technologies, and she's now currently working in Akoya Biosciences as a senior application scientist. So take it away.
Great, thank you. Can you hear me, guys? Can you hear me okay?
Yes, we can. It's all good.
Great. Good morning, everyone. I'm Niyati Jhaveri, a senior application scientist at Akoya Biosciences, the spatial biology company, and I'm coming to you live today from Menlo Park, California. I'm really excited to be here today and share some data from our newest and fastest imaging platform, which is the PhenoCycler-Fusion. We have some exciting data with our collaborator, Dr. Arutha Kulasinghe at the University of Queensland, that I'll be sharing with you today. So here there are over 300 people joining us today, either virtually or in person in Australia. And that really speaks to the excitement surrounding spatial biology right now. So what makes spatial biology so promising and so powerful? Most conventional methods for protein or gene analysis have focused on bulk methods where the tissue is either ground up or dissociated into single cells. Single cells can also be sorted by techniques such as flow cytometry to infer analytical information. But in all these cases, there's a very crucial component of spatial context that is missing. And while conventional immunohistochemistry methods can overcome this limitation, IHC is also limited by the number of targets that you can look at simultaneously. And this is really where Akoya comes in. We're really invested in enabling new dimensions of spatial biology research in terms of the number of targets as well as the type of analytes that can be looked at together. So we believe in a multi-omic as well as a multiplexed approach to inferring tissue biology. So most of you must have heard the term no cell is an island. Tissues are very complex. They're made up of a multitude of different cell types. The organization of these cells is critical to the functioning of normal tissues. And we're finding more and more that the disorganization and disruption of this tissue architecture plays an important role in the tumorigenic pathogenesis and progression process. So spatial biology, with the power of single cell resolution and whole slide imaging, really provides a valuable tool for unbiased analysis of both tissue form and function. So Akoya started in 2017 as a 3-person company. We acquired technology that came out of Gary Nolan's lab at Stanford. Gary's a pioneer in the spatial biology field and developed the Codex co-detection indexing methodology. Akoya also acquired high-resolution imaging technology, and most of you may be familiar with the Polaris PhenoImager high-throughput imaging platform. And we've been rapidly growing from a 3-person company to over 300 employees worldwide now, the largest instrument base in the field, as well as over 450 publications in the field of spatial biology. And what makes our technology stack especially exciting is that we have platforms spanning all the way from the discovery space with the PhenoCycler, all the way to the clinical realm with the PhenoImager HT. Our newest product, the PhenoImager-Fusion, merges the best of both of these worlds with a tunable workflow that allows for high-plex imaging as well as high throughput. So we have a little video here that talks about how the Fusion system works.
Over the years, Akoya has always aimed to break new ground with innovative technologies. Now, with the integration of 2 platforms, a new era in spatial biology begins. Introducing the PhenoCycler-Fusion system, providing the speed and accuracy required for unbiased, whole slide spatial discovery. By combining automated fluidic cycling and high-speed imaging, this novel system delivers an end-to-end workflow for ultra-high-plex spatial phenotyping, a workflow that allows for walk-away automation during target labeling, tissue focusing, scanning, and data processing. The PhenoCycler multiplexing platform uses fluorescently conjugated barcodes to label the antibody-stained tissue. Through a cyclical process of reveal, image, remove, 3 biomarkers are detected during each fluorescent scan, resulting in a series of image files that are layered into a single QP TIFF file for analysis. To label the tissue prior to each scanning cycle, the PhenoCycler delivers the bar-coded reporters through a uniquely designed flow cell. The flow cell allows for staining on whole slides, so every single cell across an entire tissue section can be labeled for true, unbiased discovery and in situ spatial phenotyping. While the PhenoCycler allows for automated high-plex staining, cutting-edge slide scanning technology removes any manual intervention during focusing and image acquisition, delivering the speed, reliability, and reproducibility to accelerate your research. Following target labeling, the Fusion goes through a series of automated steps to detect the sample and ensure that the entire tissue is in focus. After imaging the sample label, rapid measurement of the sample height is determined using height sensing optics technology, or HSO. HSO enables automated focus mapping, providing an alternative to Z-stacking, improving speed and reducing data size. HSO is a patent-pending technology that measures the height of the sample to allow estimation of the focus depth across the flow cell. A near-infrared beam of light projects a pattern beam onto the sample at different Z-planes. The Z-plane, where the reflection of the pattern from the glass cover slip is sharply focused, indicates the height of the sample. This measurement is repeated at locations across the slide for accurate positioning in the Z-plane. Using the HSO-determined height, a rapid fluorescence flying overview is performed to detect the tissue sample area. Then, fine-resolution focus measurements are required at a series of X and Y locations across the tissue, generating a focus map for optimal focus during scanning. Throughout image acquisition, the stage moves seamlessly through nanometer precision coordination, capturing a multiplexed fluorescent image at each adjacent position. In-flight parallel processing tiles each image in real time, actively rendering the entire tissue image. This scanning process repeats itself after each cycle, generating a series of image layers that are assembled into a single manageable QP TIFF file, requiring only a few gigabytes of data storage rather than terabytes. Within minutes, you have analysis-ready images and can perform a comprehensive set of spatial phenotyping analyses. Scale up your spatial biology discoveries today with the PhenoCycler-Fusion.
So with the accelerated speed that PhenoCycler-Fusion enables, we can now do an ultra-high-plex experiment of over 100 biomarkers. If you're interested more in the translational and clinical space, then you can also run 100 slides per week with up to 8 biomarkers. Apart from the ultra-high-plex capabilities, as well as the single-cell and subcellular resolution, whole slide imaging with Fusion also ensures that every cell in the tissue is accounted for. Earlier this year, we also announced a collaboration with ACD to automate the RNA-scope in situ hybridization workflow for the analysis of RNA, 12 RNA targets on Fusion. And in a couple of weeks at AGBT, we will be unveiling our own internal RNA chemistry for high-plexed spatial transcriptomics. So to delve more into how the technology works, as I said, it's based on the Codex technology that was developed by Gary Nolan's lab at Stanford. Antibodies are conjugated to oligo barcodes, and tissues are stained in a multiplex panel in a single staining step. This is followed by repeated cycles of reporter hybridization, imaging, and dehybridization to reveal 3 markers at a time. And this process is automated on the PhenoCycler-Fusion platform to have a streamlined workflow from staining all the way to analysis. So there are 3 main components to this. The first is the reagent component, which includes the barcoded antibodies. We have validated antibodies for FFPE tissues as well as fresh frozen mouse tissues. We also have complementary reporters attached to fluorophore dyes and custom barcodes. So if you're working with an antibody in-house, if you're generating an antibody in-house that you're interested in combining with our validated antibodies, then we have custom barcodes available for custom conjugation. The imaging component includes the PhenoCycler-Fusion, which is the fluidics platform combined with the fastest imaging platform. And then we also have software such as Inform, as well as third-party software such as Visiopharm and QPath for analysis. So all of these are available through Akoya, and it's really an integrated end-to-end spatial biology solution. So next, I'd like to switch gears and talk about some of the different applications and research questions that you can address with this system. One of the first questions, the most basic question that you might have is, what are the different cell types in my tissue, and what is the distribution and abundance of these cell types? So this is called spatial phenotyping, mapping every tissue in the cell to create every cell in the tissue to create a tissue atlas. When you're looking at entire tissues, you might also come across rare cells, the less than 0.1% of cells. And I'll talk about a collaboration that we had with Dr. Kai Kessenbrock at UC Irvine, where we made such a discovery in breast cancer tissue. Later this year, with the multi-omic approach of both RNA and protein detection, we can also start looking into functional states of cells. And then building upon that even further, we can start to look at the organization of cells into spatial neighborhoods. This is a concept that Dr. Nolan introduced in his 2020 paper. It's a seminal paper on the importance of spatial neighborhoods in analysis, and I'll talk about this in a couple of slides as well. And then bridging the arc from discovery research all the way to the clinical realm, we can also start looking at spatial signatures, unique profiles that could have predictive value in the clinic and help us understand the basis of clinical responses. So the first one that I'd like to touch upon is spatial phenotyping. This is a human FFPE tonsil tissue that was imaged that was stained with over 23 biomarkers and imaged on the PhenoCycler-Fusion platform. All 2.5 million cells, as shown here, were imaged in less than a day. And you can really start to appreciate the complexity of the tissue architecture with the germinal centers, the lymphoid follicles, and the epithelium. With bioinformatics analysis, we were able to identify 13 unique phenotypes with differing cell expression patterns, and also look at quantitative information into the distribution as well as the abundance of these cell types. This data set is available publicly. It's an interactive data set at the link below, and I would highly encourage you to visit it and play around with it. So the next application is rare cell discovery. This is a collaboration that we had with Dr. Kai Kessenbrock. He's interested in studying the etiology of breast cancers and deciphering all the different components that make up the tumor. All breast cancers arise from lobular epithelial cells, but there's very little known about the heterogeneity within this epithelial component. And immunohistochemistry and single-cell RNA-seq really lack the depth and the spatial information needed to tackle the heterogeneity. And so with Kai, we developed a 36-plex breast tissue panel, and this is one of the images that came out from that study. It's a tissue that has over 63,000 cells, and using the 36-plex panel, we were able to identify 12 distinct phenotypes, including basal and luminal epithelial cells. What's interesting is we also found this third rare epithelial population right here. It's about 44 cells in the entire 60,000 tissue, and it would have been missed with ROI-based methods. But with the power of whole slide single-cell imaging, we were able to discover this rare population of luminal cells that has a very unique profile and may play an important role in tumor progression. So along with Kai, this technology has been widely adopted by the scientific community globally in projects such as HubMap and HCA. HubMap has over 600 publicly available data sets, and we're very proud that the largest spatial data set in cell atlasing was acquired on the PhenoCycler platform. The next application that I'd like to touch upon is spatial neighborhood analysis. So this is a very seminal paper that came out of Gary Nolan's lab in Stanford. Gary was interested in studying cutaneous T-cell lymphoma, and patients with CTCL that are on anti-PD-1 therapy have a very mixed response. So his lab was really interested in studying the mechanistic basis of this mixed response. So they started with looking at PD-L1, CD4, FOXP3 with single-plex IHC staining, and surprisingly, they found no statistically significant differences between the responding population as well as the non-responders. They also did whole exome sequencing and mass cytometry, and none of these methods really revealed any differences between the responders and the non-responders. And so they realized they had to dig deeper, and they developed a 56-plex immuno-oncology panel and started to look at the organization of these different cell types within the tissue. And that's when they got some really interesting findings. They found 3 cellular neighborhoods that were differentially expressed between the responders and the non-responders. Cellular neighborhood 5, comprised of tumor and dendritic cells, and cellular neighborhood 8, which is enriched for tumor and CD4 T cells, was highly expressed in the responding group post-treatment. And this suggests that the immune response driven in this group of patients might be driven through the CD4 T cell population. Interestingly, they also found that cellular neighborhood 10, which is an immunosuppressive T regulatory population, was enriched in the non-responding group post-treatment. And so this really explained how it's important to not just look at single-plex markers alone, but look at it more in its entirety and have a more integrated approach to understanding tumor biology. Since the paper came out in 2020, this concept of spatial neighborhood analysis has really taken off, and a lot of researchers are now incorporating this into their analysis workflows. This is just a sampling of some of the papers coming out in the last couple of months. So how does fusion fit into this arch that I just showed you? So far, we've been limited in our pace of discoveries by technologies that require a lot of investment of time, and they're limited in their plex level. But fusion strives to solve that with speed, whole slide access, high resolution data that doesn't require a lot of storage capacity, as well as multi-omic capabilities with both RNA and protein detection. So with fusion and the accelerated speed, we can now start looking at more number of markers, which means more spatial phenotypes that could be discovered. We can also look at larger areas of tissue, and this could lead to an increased potential of more rare cell discoveries. This could also translate to more spatial neighborhoods and ultimately to more unique spatial signatures that could have prognostic value in the clinic. I've been working with this system for 6 months now since I joined Akoya, and I'd like to talk in the next couple of slides about a project that I've been working on. It's called the PhenoCycler project, and the inspiration for this project really came from the seminal Hallmarks of Cancer paper by Doug Hanahan and Bob Weinberg in the early 2000s. So based on the hallmarks of cancer and different proteins that are involved in different pathways involved in cancer, we curated this 100-plus plex panel, which has different modules, each revealing information on different hallmarks of cancer. So we have 10 modules here. Each of the little squares here highlights the hallmark of cancer that it reveals. And when combined together in a single ultra-high-plex experiment, this really provides an integrated overview of the landscape of cancer progression. Data set coming out of this is in human FFPE tonsil tissue. This is the largest single-cell spatial phenotyping data set with over 2.25 million cells and 103 markers that were imaged in less than 2 days. So you can really see the wealth of information here as you cycle through the different markers and the different hallmarks of cancer, starting from the immune microenvironment to angiogenesis, invasion, tissue structure markers, cellular metabolomics, proliferation, and DNA damage and stress and death pathways. And on the right here, we have a bioinformatics team that performed unbiased clustering, hierarchical clustering analysis on this data set to reveal how all of these different markers correlate with each other. So there's really a wealth of data here, and you can see that we have like different functional groups here grouping together. We have the T cell markers, CD3E, CD4, STAB70, which is a transcriptional factor associated with T cell. We see 2 different populations of epithelial cells, which speaks to the heterogeneity within this component. We also see different subsets like B cells, macrophages, and vascular markers grouping together. So this not only validates our panel, but it also provides a depth of information like never before. So having validated the panel in tonsil tissue, we next applied it to head and neck cancer. This is really an exquisite piece of tissue that we received from our collaborator, Dr. Arutha Kulasinghe at the University of Queensland. And this video is just cycling through all 103 markers to show the complexity and the complex topography of this tissue. So what's really interesting about this sample is that, is up here on the top left, we have the normal squamous epithelium, and then we have these two pieces of tumor tissue. And we can really start to appreciate the disruption of normal architecture that occurs along with tumorigenesis. Head and neck cancers are a group of cancers originating in different sites, as shown here, and only about 15 to 20% of head and neck patients respond to immunotherapy. So with Dr. Arutha Kulasinghe, we're interested in studying how the spatial phenotype in these tissues correlates with the clinical response. This is another really interesting sample that we received from him, and you can see from the H&E image, this is the normal lymphoid region where you have the germinal centers, you have the squamous epithelium here, and then based on the pathologist's notes, this region jutting out here was annotated as the tumor. But when we ran the 100-plex panel on this tissue, we found that there's more to the tumor than just that piece. So this is a sampling of markers, T-cell markers, as well as epithelial markers, including pan-CK. And we can see the CD4 cells in the normal lymphoid region. We can see the germinal centers here. We can see the high levels of CD8 infiltration in the tumor, as well as the dense epithelial outgrowth. Apart from the dense epithelial outgrowth in the tumor, we also see massive amounts of epithelial growth in these other tumor regions. And indeed, when our bioinformatics team did deep analysis on these tissues, they found that there are 3 different tumor regions in this tissue, apart from the original tumor that was annotated by the pathologist. So we have tumor 1, 2, 3, and 4 with very unique spatial neighborhoods. Analysis of the different cell lineage markers also revealed 14 broad phenotypes in this tissue, and these have all been spatially mapped back onto the tissue to reveal the complex topography. So we see the B cells here, the normal squamous epithelium, the M1 macrophages, and the unique profiles, the unique cellular profiles of the 4 tumor regions. We also have the cross-correlation analysis here, which shows the richness of the data that we got out of this, and we're still working through analyzing some of this and making sense of it all. So when we did quantitative analysis on the different cell lineages here, we also found some really interesting differences between the 4 tumor regions. And this speaks to the intratumoral heterogeneity that is present within this one patient sample. So here we have tumor 4 stained with CD68 and CD20, a B-cell marker, and we see that tumor 4 had very high levels of CD68, M1 macrophage infiltration. We also see that both tumor 2 and tumor 4 had very high levels of CD8 T-cell infiltration. So together, the CD8 and the CD68 data suggest that tumor 4 is actively being attacked by the immune system relative to the other tumors. So that was just a subset of the data looking at cell lineage markers in the immune microenvironment. But when we extended this to other markers that were present in the 100-plex panel, the metabolic pathway markers, the stress markers, we see intratumoral heterogeneity across multiple levels of tumor biology. So these are heat maps showing differences in metabolic and stress markers in the CD8 T-cell population, the macrophage population, as well as the normal epithelium and the 4 distinct tumor regions. And one particular difference that I'd like to highlight is the differential expression of glucose 6-phosphate dehydrogenase and BAC. So glucose 6-phosphate dehydrogenase is an enzyme involved in glucose metabolism. It's been implicated in tumor progression and resistance to therapies. BAC is a pro-apoptotic regulator. It's been implicated in cell death pathways. And we see this very interesting profile where tumor 3 has really high levels of G6PD, whereas tumor 1, 2, and 4 have very high levels of BAC. And if you remember the slide that I just showed you before this, tumor 4 was also the one that was being actively attacked by the immune system. It had high CD68 positive cells, it had a high CD8 positive cells, and together with the back data, this suggests that this tumor is undergoing a lot of stress and death. On the other hand, we have tumor 3 here that is actively proliferating. It has invasion markers like MMP9 and glucose 6-phosphate dehydrogenase, which plays an important role in resistance to clinical therapies. So we can really start to appreciate at the single cell level with spatial biology the unique spatial neighborhoods within just one sample of tumor. And this data really speaks to the power of whole slide single cell analysis to reveal the complex topography of human tissues. From Gary Nolan's paper, we now realize that single plex IHC alone is not sufficient to tackle a complex disease like cancer. And we really need to have a more multifaceted approach to study all the different pathways that integrate together to contribute to tumor progression and pathogenesis. So what was interesting about this sample is that when we went back to our collaborator with it and looked back at the clinical data, the patient only had a partial response to immuno-oncology therapy. And now with the 103-plex panel, we can start to decipher and uncover the mechanistic insights behind that partial response. So if you're interested in analyzing this panel in your tissue of interest, we do have a 100-plex grant program currently running. We would love to hear about your research and how we can leverage the cancer hallmarks panel to understand more about tumor biology. With that, I can take any questions you may have.
Thank you very much, Niyati, for a beautiful talk. It's mind-blowing to see that 100-plex results up here. Do we have any questions on the floor? I must start with online questions. So how many cycles of staining can be done on one sections, and does the quality of the staining or the signal detection decreases over time?
Yeah, that's a great question. So the staining is all done in a single step. For the 100-plex, it was split across 2 steps, but the staining is all done in one step, and then it's repeated cycles of reporter hybridization, imaging, and dehybridization. And we've gone up to 45 cycles without seeing any loss in signal strength or specificity. So we do a lot of validation work within our R&D team to make sure that we're maintaining that specificity and signal strength throughout the different cycles.
And a question for my own. So I was wondering, like, I would assume different target would require different antigen retrieval conditions. So how do you accommodate the differences between different targets?
Yeah, that's a great question as well. So we are very careful with the clones that we select. The panel that I showed you used TRIS-based antigen retrieval. So we made sure we selected clones that would be compatible with the antigen retrieval method that we were using.
Yes. And I was wondering also, like, I could see that there's a pipeline to do multi-omics, meaning that RNA and protein using the same platform. So does it also can be applied on the same tissue section as well, or is it on adjacent section?
Yes, so at EGBT, we'll be unveiling more about our RNA chemistry and spatial transcriptomics. And then, yes, down the road, that's the goal, to combine both RNA and protein in a single tissue.
Yes. And I've got online questions in here. So can you detect HIV or HPV at the single cell level together with gene expression data?
Yes, so if you have antibodies, if you have good antibody clones against HPV or HIV, then we can definitely combine that. We have custom barcodes that you can conjugate the antibody with. These are compatible with the currently available validated antibodies that we have in the panel. So as long as you have a good antibody, it's possible.
All right, and one final question from online, which is quite important. How much does it cost per panel on per slide?
Yeah, that's a great question. So we do have our sales manager, Jackie Mao, there on site, and she would be the best person to address that question. I believe she also has one of the interactive data sets, so it's really cool. If you get a chance, do visit her and check that out.
Yeah, I'll also encourage all the in-person attendees to mingle with our sponsors during the morning tea break and afternoon break. And with that, thank you again, Niyati, for a really wonderful talk.
Thank you.
And I'll pass on the time to Verena.